Hello, everyone. Welcome to my standard edgeR workflow video tutorial. So edgeR is one of the best package to analyze RNA sequencing data. In my previous video tutorials, I showed you how to create pseudo RNA sequencing data from single-cell RNA sequencing data. And we use the edgeR to identify differentially expressed genes. Today I'm going to use the bulk RNA sequencing data to show you the standard edgeR workflow. In this video tutorial, I use the simplified code to go through the edgeR workflow. I don't use lots of code to show the data. So first, let's load the libraries. We need tidyverse, lemma, and edgeR. So for today's demonstration, I use the same datanet in edgeR case studies. The first case study, RNA-seq of oral carcinoma against matched normal tissue. If you want to use this dataset for practice, you can download the data from PLOS ONE journal. The data is in supplementary table 1. So I downloaded the data and made some modifications for today's demonstration. And I saved this data in my edgeR folder as oral RNA CSV. So for the standard edgeR workflow, the first step is to load the data. So let's load the data in. We can click the data, have a look at the data information. You can see the first column is the gene ID. The second column is the gene symbol. Then from the third to the eighth column, we have the RNA sequencing information. So this dataset performed bulk RNA sequencing using the tissue from 3 patients. You can see the number for patients 8, 33, and 51. M means normal tissue, and T means the oral carcinoma tissue. So we loaded the data in step 1. In step 2, we can create a DGnist. In this step, you can add the tissue information into the DGnist. We know we have normal tissue and tumor tissue from 3 patients. We can set the tissue information. Then we use the count and the gene information from the dataset we just loaded in to create the DGnist. We loaded the data as oral RNA. I showed you just now the counts for the sequencing data in column 3 to column 8, and the gene information in the column 1 to column 2. Then we can add the tissue information in the group argument to create a DGnist for edgeR analysis. So let's create the DGnist now. You can see we have a large DGnist here. It has 3 elements. We can have a look at the data information in the DGnist. You can see the first element contains the count information. The second element is the samples. You can see we have the group for the tissue. You can see in the group we have the tissue information. We have normal tissue and the tumor tissue. Then we have the library size information. Here is the normalization factors. Because we haven't performed library size normalization, so at the moment, the normalization factors are 1 for each dataset. And the third element is the gene information. You can see we saved the gene ID and the gene symbol information in the DGnist. So we created the DGnist for the analysis. So the third step is to filter the genes. So we can use the filter by expression function to filter the genes. Let's perform the filtration. We can use the table to have a look at the genes. You can see in this dataset, we have 10,510 genes, but all the genes labeled as true, so we actually don't need to filter the genes. In case you have genes need to filter out, we can run this code for demonstration to filter the lowly expressed genes. So after the filtering, we can normalize the library size. This is step 4. Let's normalize the library size. So in step 5, we can export the data. This step is optional. If you don't run the plotmds function, you can still perform the rest analysis. If we run the plotmds function, we can check if there is a difference on the gene expression level between normal tissue and tumor tissue. So let's run the plotmds function, and we can zoom in. You can see we have 6 samples here. All the samples from the normal tissue were clustered on the right-hand side, and all the tumor tissues were clustered on the left-hand side. So you can clearly see the difference on gene expression level between normal tissue and the tumor sample. As I said, this step is optional, but it is very useful to look at the data. So in step 6, we can design the matrix. In this video tutorial, I try to show you the simplified edgeR workflow, and you can follow the most code to run your data. But step 6, the matrix design, is more flexible because we need to design a matrix according to your research question. So in this dataset, we want to identify differentially expressed genes between tumor tissue and normal tissue. And also we want to identify up and down-regulated genes within the same patient. So we are going to set the patient information. We have 3 patients, number 8, number 33, and number 51 for the normal and the tumor tissue. So let's set the patient's information. Then we can design the model matrix to include the patients and the tissue information. We already included the tissue information when we created the DGnist. We set the tissue information as normal tissue and the tumor tissue. So let's design the matrix. And we can have a look at the matrix. You can see in the first column is the intercept column. The second column is the patient 33. The third one is patient 51. Then the last column is the tissue information. You can see we have the normal tissue as 0, tumor tissue as 1. At the moment, the row names is 1 to 6. We can get the row names from the column names in the DGnist. Let's get the row names. You can see now we have the patient number and the tissue information in the row names. So we designed the matrix. In next step, step 7, we can estimate the dispersion. Here we can use the matrix information to estimate the dispersion. So let's run step 7. Now we can plot the biological coefficient of variance to see the estimated dispersion. Let's run. And we can zoom in to see the dispersions. You can see the tangle-wise dispersion was shown in black dots for each gene. The common dispersion is in red because the common dispersion value is the same for all the genes. So you can see a straight red line. Then the trended dispersion is in blue. So after dispersion estimation, we can perform differential expression in next step. So this is step 8. In this step, we will identify differentially expressed genes between tumor and normal samples. So edgeR has two types tests available for differential expression analysis. So the first one is the quasi-likelihood F-test, and the second one is the likelihood ratio test. So I'm going to show you both tests, and later we will compare the gene difference between the F-test and the likelihood ratio test. So first we can perform the quasi-likelihood F-test. Before we perform the F-test, we need to fit the gene-wide negative binomial generalized linear model for quasi-likelihood test. So let's fit the model first. Now we can perform the quasi-likelihood F-test. You can see we named the test result as QLF. It is the formal class DGE likelihood ratio test object. We can click the data and have a look. You can see here are all the information about the test. But for us, normally we want to see the table because the table contains the information for the log FC values, log CPM values, the value for the F-test, and also the p-values. So edgeR has a function called top text to look at the top genes that show significantly difference. We can run this function. You can set the number you want to see. For example, here we are going to look at the top 20 genes. Let's run. You can see down here we have 20 genes show significantly difference. You can see the first column is the gene ID, second column is the gene symbol. Then we have the log FC changes, log CPM value, F-test, p-value, and the FDR value for false discovery rate. So we know those information for all the genes are in the table data slot. So we can get the data from the table data slot. We named as A here. Then you can see here we have a data frame contains 10,510 genes. And we can click the table, have a look. You can see the log FC, log CPMF value, p-values for all the genes. But we know not all the genes have a significant difference between the normal tissue and the tumor tissue. So we can filter the genes to keep the genes that show significantly difference. Let's run. You can see now we only have 2080 genes show significantly difference between tumor and normal tissue. If you want, you can save the dataset for the genes that show significantly difference. Let's run. So we performed the quasi-likelihood F-test. Next, I can show you how to perform the likelihood ratio test. So first we can fit the gene-wide negative binomial generalized linear model. Let's run. Then next we can perform the likelihood ratio test. Again, we can use the top text function to look at the top genes show significantly difference. Here we set the number as 10. Let's run. You can see here we show 10 genes that show significantly difference between normal tissue and tumor tissue. Once again, we can get the table for all the genes. You can see at the moment we have the values for all the genes. Then we can filter the genes and just keep the genes that show significantly difference. We can run. Now you can see in B we got 2,286 genes. So between two tests, you can see in the likelihood ratio test, we identified more genes that show significantly difference. It is about 206 genes. So again, you can save the data. Let's run. So I already showed you that the gene numbers are slightly different between two tests. We can get the gene names from the F-test, then gene names from the likelihood ratio test. Then we can use the intersect function to look at how many common genes that we identified from both tests. So let's run the intersect function. You can see in the C, we have 2,066 genes. And those genes were identified by both F-test and the likelihood ratio test. So you can see both tests worked really well. So it will be your choice to use which test for your dataset. So next we can use the likelihood ratio test as an example to summarize the genes that show up and down regulation. First, we can order the p-values for all the genes in the likelihood ratio test. Let's run. And then we can use the decide test function and summarize the gene numbers. We can run. You can see from the likelihood ratio test, we identified 938 genes that were downregulated in tumor tissue and 9,241 genes. They don't show significant changes, and we identified 331 genes that show upregulation. And also we can use the count per million function to look at the gene expression level for the top 10 genes. We can run. You can see the first gene is highly expressed in the normal tissue, then expression is very low in the tumor tissue. You can see the same pattern in the other two patients. Then the second gene, you can see it has no expression in normal tissue, then it is upregulated in the tumor tissue. Once again, you can see the same pattern for this gene expression in patients 33 and patients 51. So we summarized the gene numbers that show up and down regulations. We can make a plot to visualize the gene numbers that show up regulation and down regulation. Again, we are going to use the likelihood ratio test as the example. Let's run the plot. And we can add the lines to log-fold changes as twofold down regulation and twofold up regulation. Let's run. And we can zoom in. You can see upregulated genes in red here, and the downregulated genes are labeled in blue. And the black dot show the genes that don't have significant changes between tumor tissue and the normal tissue. So we run the edgeR standard workflow and identify the genes that show significantly changes. In the final step, I'm going to show you how to perform pathway analysis because the edgeR package incorporated gene oncology pathway analysis and also the KEGG pathway analysis. First, we can run the gene oncology analysis. So it is very simple here. We can use the likelihood ratio test as the example to run the goal analysis. Let's run. You can see in the goal analysis we have 20,146 observations. We can use the top goal function to look at the top 30 signaling pathways. We can run. You can see here are the top 30 signaling pathways from the goal analysis. And next, we can run the KEGG pathway analysis. So if you run KEGG pathway analysis, you do need the internet connection for this analysis. You can see it is very quick. And you can see from the KEG data frame, we identified 356 pathways from the KEGG analysis. Again, we can use the top KEGG function to look at the top signaling pathways. Let's run. You can see here the signaling pathways that are activated in the tumor samples. Okay, so that's the full tutorial for the standard edgeR workflow. I simplified the code. Then if you can just run through those code, you can perform your analysis for your dataset. I hope this video could help your bulk RNA-seq data analysis. Please subscribe my channel if you haven't to do so, and share my videos with your friends. Thank you.